Hi, thanks for being
Hello,
here. Hey.
Katarina.
Hi, Vlatko. Hope I'm saying your name right. Hi, Cecile.
Hi, Katarina. Hi, Victoria. Hi, Biatko.
Hello, hello.
Hi, can you hear me? Hi, hello, can you hear me?
Yes, very well.
It's Vladko speaking. Hi.
Thanks so much for making it. I'm so glad.
Yeah, I think I'm a bit of a Luddite, like I said, but I did manage. I'm very happy.
Yeah, we are happy, very happy that you made it. So thank you for making
Thanks a
time.
lot for inviting us. It's a pleasure.
We'll wait. The room is scheduled for 4, so we'll wait a few minutes for people to come in. So yeah,
So,
then I'll introduce you. Thanks.
Katarina, I was thinking I could move myself down to the audience until the actual presenters, so you could mod the presenters so that the lineup will represent the calendar.
Oh, you're included in the calendar. You're totally fine. It's good. All good. Thank
Sure.
you. Hi, Rainer. How are you? Thanks for coming. Thank you for being here. Can you hear us okay?
Yeah, I'm trying to ping some people.
Yeah, me too. I did the same. Yeah, we can start to, with introductions, and yeah, and then I'll give you to the stage. So yeah, welcome everyone to the Science Society Club, and we are very honored to have guest speakers here that will talk about their very interesting and highly discussed research. And yeah, I'll start introducing you Dr. Rainer Dumke. He is an associate professor at in Singapore, and he did his PhD at the Leibniz University Hannover, and he did his postdoc at Max Planck Institute for the Science of Light. And he now is at the Center for Quantum Technologies at the National University of Singapore. And yeah, he won the IG Nobel Prize in 2019. And for people that don't know the IG Nobel Prize, it is the Joker younger brother of the highly prestigious Nobel Prize, and it was awarded for the research over magnetized cockroaches. So that's a very cool prize to win. And yeah, in his group, they are working on further development of technologies with the aim of using quantum nature of ultra cold atomic matter. And their projects include to work with superconducting atom chips, Bose-Einstein condensates, miniaturized optical systems for quantum information processing, and building a compact and transportable atom interferometer for the precise determination of local gravity. Yeah, and then we have also Professor Dr. Vlatko Vedal. He is professor of quantum information science at the University of Oxford and governing body fellow of Wolfson College. And he's also principal investigator in Singapore at CQT, professor of physics, and Centauri Professor of Quantum Information at Leeds University. And he did his PhD at the Imperial College of Science, Technology and Medicine, and his Bachelor in Physics at the Imperial College of Science. And he won also a lot of awards throughout his career. So yeah, we are very honored to have you both here to introduce us to your very interesting and exciting work. Thank you so much for coming. Rainer, can you hear us? Vlatko, would you like to start until Rainer maybe is having trouble with the app? I'm not sure.
Of
Um,
course. Can you hear me?
yes.
I'm happy to start.
Okay, Vlatko, can I interrupt just one moment? I want to share this for Rainer. Rainer, it's possible if you go off of your Wi-Fi that you would be able to get reception if that's the problem, and just want to make sure that you know where your mic is. And also another thing you can try is leave the room and then come back in, and that could also help to enable your mic. Okay, Vlatko, excuse me. The mic is yours.
Not at all. Actually, it's thank you for inviting me and for your very glamorous introduction. Um, I just want to kind of maybe comment on some of the research that you mentioned that we've been doing. I think the exciting question, and please interrupt me if there are any questions or clarifications. The exciting thing for us is really to understand whether quantum mechanics applies to anything, any object in the universe. We know that quantum mechanics works really well for small objects. You know, if you're talking about atoms and subatomic particles and light and, you know, particles of light, photons and all of that, we've tested these things quite extensively. However, we haven't tested quantum mechanics in the macroscopic domain. The question is, if you go larger and larger, if you go into more complex molecules, the question is whether quantum mechanics is still valid at that level. Are there quantum processes that are really relevant for complicated chemical processes that are relevant for biological functions? And I think this is a completely unexplored direction. And it's very exciting, I think, because ultimately, what we are really trying to understand is whether quantum physics is relevant for living systems. So that really sounds like a really fantastic question, because it involves not just physics, it involves chemistry and understanding various issues in biology. So it's kind of a multidisciplinary direction. It crosses boundaries between many fields. Also, computing, I think, is something that you're going to hear about, because ultimately Reiner's experiment was really about a hybrid technology where he was coupling a living system to a quantum bit inside his laboratory, a specific quantum bit that we can also talk more about, and I'm happy to explain it. So I think the exciting thing for us really was to go into this macroscopic direction. That's the main thing. Does quantum mechanics really apply at that level of complexity?
Sorry, can you hear me now?
Yeah, we can hear you, but it's a little faint.
Oh, okay, perfect. Yeah, anyways, please go ahead, Radko. And yeah, I can jump in anytime.
I just wanted to set the scene, Rainer. You can maybe go and explain the experiment a little bit, because I think the big vision really for us, like I said, is to take quantum mechanics from this microscopic domain of small objects, of a handful of atoms and small molecules and photons and so on, and really test it on something big. And I think this was exciting for us because there are various claims, even in physics, that quantum mechanics will actually cease to be applicable. It will collapse, if you like, when it encounters a big enough complex object. And certainly living systems are, as far as quantum physics is concerned, big and complicated. So there are even some suggestions, some hypotheses in physics that the quantum mechanics will actually not be applicable to living systems. And I think this is really one of our key driving motivations. And maybe, Rainer, you can actually say now a bit more about the experiment. I think if this vision is clear, then I think maybe the next step logically is the experiment.
Yes, absolutely. So yeah, as you have mentioned, this was our driving idea. So, however, let's say having this idea, and the question is actually now, how can we actually complete it, right, or do this task? And actually to let's say couple a quantum mechanical system with, let's say, a living system, that has certain difficulties, right? Because let's say one has to be able to adopt, let's say, a quantum mechanical system to, let's say, an environment which biological systems like, right? So it's typically then at room temperature, 300 Kelvin, and the environment is actually noisy and wet. So let's say one way is, let's say, to try to change let's say, quantum system that can actually work in this type of environment. So it is, let's say, one has to use then, let's say, higher energies, typically optical frequencies, and also very, very short timescales. Or the other option is actually to find an organism which actually survives in a surrounding which a quantum system likes, meaning at low energy, and also, let's say, in a surrounding which is non-interacting, so which actually means the system has to be inside a vacuum. And there are actually not so many organisms which would be able to survive being exposed to such an environment. In fact, this organism has to undergo something like a cryptobiosis, in a sort of, which is a latent state of life, which, let's say, some life forms can actually enter when they are, let's say, in response to adverse environmental conditions, for example, like freezing or being in a vacuum. And let's say in this, let's say, state, all measurable metabolic processes come to actually to a standstill. And then when actually the environment returns to normal, the organism actually goes then again back to normal. And the best life form for our experiments which can actually do this, also in terms of size and ability to sustain these conditions, are actually tardigrade or tardigrades. In particular, actually, a species or specimen of a Danish population, which we have actually used, and actually this particular species is also very robust, very robust, and can actually sustain harsh environments. When this tardigrade actually undergoes this cryptobiosis, it will reduce its volume, so it's shrinking and curling up, and goes into this metabolic state, which is actually known as the TAN state. This tardigrade was actually collected by one of our collaborators, Nadia. She was actually instrumental in, let's say, taming the tardigrade, and actually she knew how to handle this. This was collected from a roof gutter in Denmark and then prepared in the TAN state, so that, let's say, the tardigrade could survive the journey from Denmark to Singapore, where we have done, let's say, all these experiments. When the tardigrade then arrived in Singapore, we placed the tardigrade on the superconducting qubit. The superconducting qubit, one could actually imagine it's some microfabricated system on a silicon wafer. For the qubit which we have using, we used a transpond qubit. This transpond qubit is actually made out of, let's say, principal big capacitor and a Josephson junction. The capacitor is, let's say, almost mesoscopic, so you can see it with your bare eyes, and it's actually ideal to use, let's say, this capacitor to couple to the tardigrade itself, which we actually put directly in between this capacitor electrodes. Then, let's say, the qubit is placed inside a 3D aluminum cavity, which is superconducting at the temperature in the dilution refrigerator, which we also use actually to read out the qubit state. Then the whole sample is placed into a dilution refrigerator and cooled down to about 10 millikelvin, where we also then start the measurements. At the same time, of course, the system is also evacuated to pressures below 10 to minus 6 millibars. And what we have seen is actually that the, let's say, resonance frequency of the qubit is shifted in presence of the tardigrade. So the question is, how can we understand this? So there's, let's say, a microscopic picture as following. The tardigrade, as I said before, is placed between this capacitor electrons of the transmon qubit. And then if the transmon is, let's say, in the state 0, so in the ground state, nothing is happening. However, if you promote the, let's say, transmon qubit to an excited state, let's say with a microwave pulse, then the electric field inside the transmon qubit is actually oscillating between the also between the capacitor electrodes, and this oscillating field is also interacting with the molecules inside the tardigrade and polarizes them, which leads to an effective change of the electric field, and with this also a change in the resonance frequency of the combined tardigrade qubit system. It's maybe also worthwhile to note that if you look actually at, let's say, the coherence time of, let's say, only the qubit alone and the qubit tardigrade system, they are roughly the same. Meaning actually that the, let's say, qubit tardigrade system is well isolated and doesn't cover to an environment. So it's a well isolated quantum system. So if you, let's say, describe the wave function, it spreads across, let's say, the whole system, across the organic qubit and the tardigrade itself. After this experiment, we have done, let's say, a second experiment where we coupled the qubit tardigrade system to another qubit, which was also located in the cavity. And now we have actually this two systems, the qubit tardigrade system and the qubit, and we could actually also, or we could actually entangle these two systems also with each other. So we made actually a base state with a fidelity of 82%. The amazing part, in fact, of the experiment, which, let's say, especially we as physicists found pretty breathtaking, was that after finishing the experiment, which took about 420 hours, we gently increased again the pressure and the temperature of the dilution refrigerator to ambient condition, took out the tardigrade from the qubit system and reintroduced it to the, let's say, normal living conditions. So put it inside a container with some food. And then after a short while, we have actually seen that the tardigrade left the TAN state. So so before it was curled up, so it went to the normal state and actually enjoyed the food which we have provided. So we could really see how it was clamping with its little feet on the food and munching it away. So was living. However, of course, it's maybe also worthwhile to note that we have done this experiment quite a few times, and especially in the beginning, not many tardigrades actually survived. So there were only a few which have survived, and one, the let's say main reason which we found is that actually the heating up and introducing again pressure. How this is actually done has to be very gently and very, very slowly, so that the tardigrade can actually survive this waking up procedure again. Question, of course, is now to say, okay, were the tardigrades alive during the experiment? What we can say is they were alive before and they were alive after the experiments. During the experiments, they were in this latent life form, so which is this unmetabolic state. And we could actually, well, one could actually say that, let's say, during the experiment, there was actually no metabolic activities present in the tardigrade itself, because it was at this very low temperatures, where one could say that the molecules are in their ground state, or there are also no chemical reactions happening at this low temperatures because the energy is not enough. One could, from a physics point of view, one could actually model the tardigrade as, let's say, just a sphere as a polarizable sphere. Right. So was the tardigrade entangled with the qubit? I would say actually yes, but we actually could not measure the entanglement itself on the tardigrade, only on the coupled systems. But maybe actually also Vlatko can jump in here a little bit and discuss a little bit about the entanglement of the, let's say, qubit with the tardigrade and how to actually interpret this.
Rainer, I'm very happy to jump in unless there are some questions and comments from your discussion so far. But I'm happy to talk a bit about entanglement as well.
Yes, please go ahead. Thank
All
you.
right, excellent. Okay, so let me maybe take a step back again and say what is really exciting to us in this particular experiment. If we go back, I think everyone would have heard about this famous thought experiment, and you know, we are hoping that what Rainer did makes Schrodinger's thought experiment a little bit less of a thought experiment and a little bit more of reality. Schrodinger had this vision back in 1935. He had the vision that that it's not acceptable to say that quantum mechanics is weird, but that's okay because it only applies to small things that are kind of irrelevant in the everyday world. And he came up with this famous cat experiment, right? The Schrodinger cat experiment. And the point he was demonstrating there is that the key feature of quantum mechanics is that quantum things can be in many different states at the same time. So, you know, the superconducting qubit that Rainer was describing, you could literally have electrons which exist in two different positions at the same time. You can have a certain number of electrons in one location in this qubit, and you can have it at the same time in another location. This is this famous superposition in quantum mechanics, and that's why we call it a quantum bit, not just a classical bit. Now Schrodinger was thinking, what happens if this kind of quantum object interacts with something else? In his case, he chose this famous cat. I have no idea why a cat. I actually prefer cats to dogs. I would have probably done this with a dog or with something else. But anyway, he chose a cat, and certainly, you know, it's one of the more advanced forms of life, and I guess that's what he wanted to illustrate. And then he said, well, look, if this atom in my case has either decayed or not decayed, but actually this is simultaneously present at the same time there, then when the cat looks at it, in one branch of this state, if you like, it's going to be okay because the atom has not decayed. But in the other branch, the atom decays, and this decay breaks a bottle with poison that actually poisons the cat. So he said, if I take quantum mechanics seriously, and if I think it applies to cats as much as it applies to atoms, then I've got to conclude that there are simultaneously two states of the cat. One is being alive and the other one is being dead. So he said this tiny superposition of an atom emitting a photon or not emitting a photon, when the cat couples to it, suddenly leads to a seemingly paradoxical state where the cat is dead and alive at the same time. How can that be? And you know, Schrodinger initially thought that this was somewhat of a paradox potentially. But I think as he continued with his research and towards the end of his life, he in fact changed his mind and he thought that's how things are. You know, this is the one of the implications of taking quantum physics seriously. And in fact, what Rainer has done, and this is probably just only a first experiment in that direction, is to really try to see what happens if you take admittedly a far simpler living system than a cat, and you try to couple it to a quantum bit that's really in a superposition of two different, almost what you would call very distinguishable, in fact, states of electrons. Which state is the tardigrade going to couple to? And as Rainer said, if you take quantum mechanics seriously, and if you think that it applies to whatever the molecules that are relevant inside the tardigrade would be in this case, then the inescapable conclusion really is that the tardigrade becomes quantum entangled to this superconducting qubit. Of course, it's not as dramatic as the tardigrade being dead and alive, because we don't really understand yet what kind of degrees of freedom this would be. But it really is fascinating and amazing that it is a kind of macroscopic living system, being able to somehow respond to a quantum superposition and join this entangled state. And I think what Rainer mentioned is that the two crucial bits of evidence that this really is so. I mean, you can of course question this, and we can do more detailed analysis, both theoretically, and experiments can probe this more. And I think that's exactly what Rainer and I have been discussing. In fact, how to do more kind of detailed experiments and more sophisticated experiments. But I think two crucial bits of evidence is that if you were to measure this new hybrid system, the entangled system between the qubit and the tardigrade, in fact, it behaves very much like the qubit itself. It hasn't lost any of its quantumness, if you like, quantum ability, due to being entangled. And so that's a very important thing to say. It's not that the tardigrade acts as a noisy system, as a destructive system. It in fact joins the qubit in much the same way to create yet another qubit, if you like. And on top of it, then Rainer was describing the next experiment, which is to bring yet another qubit, a fresh superconducting qubit, and in fact show that even this qubit can form yet another entangled. So this would be like a triple entangled state where you've got two superconducting qubits, somehow both coupled, both entangled quantum mechanically to this tardigrade. So these are the kind of two bits of evidence that very strongly, I would say, suggest to us that there is really a quantum entangled state there. Now, what Rainer did allude to, and I think this is by all means very much worth doing, and I think it should be one of our next steps, is to really try to probe this organism independently of the qubit and try to really zoom into it and go inside and try to identify possibly which particular molecules inside the tardigrade. It's unlikely that the whole tardigrade responds to this. There's always going to be some part of the tardigrade, some kind of charge distributed inside the tardigrade that's going to get entangled to this superconducting qubit. And of course, a big question for us, and we have no idea, and in fact, even our colleagues in biology don't really understand this well, is which particular part of the tardigrade really is electrically responding to this superposed quantum bit. I think this would be a great question. And looking beyond that, what really is exciting is, can we do even better than that? Could we entangle even more living systems? Could we make them more complicated? Could we make them more responsive while they're being kind of quantum mechanically manipulated and probed? And there are huge, like I said, you should really think of this experiment as maybe the first of its kind, but we can really see many follow-ups that would be even more exciting, and that would really allow us to probe this question of universal quantumness, if you like, much more deeply than what we are doing now. I don't know, Rainer, if you want to add anything to this as well.
No, I think that captures actually the main point very nicely. And yes, I also agree that, let's say, we hope that, let's say, with this experiments which we have done, and also the theory which we have developed, or which we have developed around the experiment, that this is just the trigger point for actually more exciting experiments to actually come, to actually really, let's say, think now, uh, what can we do better? What can we do with the next steps? Finding actually, as you said, molecules within the tardigrade, which you can really try to, let's say, latch on and measure directly when it's actually entangling with the organizing qubits, or are there actually even other possibilities? Interesting would be also to see maybe there are, let's say, also other, let's say, living systems which we can actually use. So yeah, these are actually the next challenges which are on the horizon.
If I may say,
Um,
we had here maybe a month or 2 months ago, Cecile. We had Professor Woodward from Tokyo University here, and he talked about his research where he saw that a magnetic field changed fluorescent signal in human cells, like intrinsic fluorescent signal. And he showed that spin correlated radical pairs I just read, are remarkable entities that are intermediates in a range of chemical and biological reactions due to their very special quantum mechanical properties that show reactivity that can be influenced by even very weak magnetic fields. So I don't know if it would be cool to collaborate with him or troubleshoot with him, because he might have an idea in the biological system what could, to like pin, like narrow it down at what molecules to look at.
yeah, I know his work. So it's very nice. The experiment he has done has actually shown that I think it was with flavorine or so, some large molecule, where actually the, let's say, reaction inside the molecule depends on the applied magnetic field strength. We have actually done, let's say, similar experiments, or let's say going in a similar direction, also looking at magnetoreceptions of animals, with actually this American cockroaches, which we've done a couple of years ago. And there, what we have, let's say, looked at was if, let's say, in this case, this biological system, this American cockroach, is actually susceptible to ambient magnetic fields. So magnetic fields of about half a Gauss, which is extremely, let's say, low magnetic fields to be, let's say, detected for a system at 300 Kelvin. Of course, you can, let's say, think with a compass, I can actually see it. But let's say making a compass is also there. Actually, the needle has to be macroscopic, and it has to be also, let's say, housed, let's say, in a non-disturbing environment. So anyways, we have done, let's say, these experiments with the American cockroach. We have done some behavior experiments. We have seen that this cockroaches actually change their behavior by, let's say, applying this, let's say, magnetic field of about half a Gauss. You could also measure with using atomic magnetometer the magnetization of the American cockroach, and actually have seen then that, let's say, the magnetic response, which actually would probe, let's say, a classical mechanism for measuring, so a classical mechanism which the cockroach would actually use to measure a magnetic field. We have actually shown that this classical mechanism would actually not or could not explain the behavioral response of the cockroaches. And then we have done some simulations and put in, let's say, this quantum mechanical system, this radical pair mechanism. And there we have seen that this might be able to explain. But we couldn't do this, let's say, experiment directly with the molecule at this point in time.
but yeah, there are, let's say, certain processes in animals, like for instance, measuring magnetic fields, which are believed that they are using, let's say, quantum mechanical processes. But also again here, one really has to, let's say, zoom in and see this, or let's say really investigate this process in much more detail.
I just want to ask a question. So, are you assuming that all the molecules or atoms in the tardigrade are coherent and in a coherent state or kind of some kind of approximate coherent state in the state that you're measuring that supposedly being in an entanglement?
Okay, Vlatko, do you want to answer or should I?
Please go ahead.
Okay, sure. So if I understand the question correctly, the question was if we assume that let's say all the let's say molecules, atoms in the tardigrade are in some kind of coherent state and let's say entangled with the superconducting qubit.
So let's say let's say we have, let's say, brought the tardigrade to this, let's say, extremely low temperatures, about 10 millikelvin. And at these temperatures, one could actually say that the molecules are in their ground state. So they are, let's say, in a, let's say, very well defined quantum mechanical state. This tardigrade is actually placed inside the, let's say, inside or between this capacitor plates of this transform qubit. And let's say this transform qubit is, let's say, printed on a let's say on a 2D surface on a silicon wafer. If you look actually the electric field of this, let's say, capacitor plates, they're actually fringe fields which are leaving the surface of the silicon wafer, right? But let's say a few, let's say tens of micrometers higher, roughly. And let's say the molecules within this volume of this fringe fields see actually the electric field from the from the superconducting qubit. So meaning, let's say the molecules in the tardigrade seeing this field from this two let's say capacitor plate from the transform qubit interact, let's say, with the transform qubits, and with this are, let's say, entangled. It's not actually the whole tardigrade itself; it's a small subfraction in the volume of the tardigrade. I hope that
So you mean
answer.
you mean like somewhere in the surface on the surface of the tardigrade that are entangled with the two qubits.
Let's say it's not necessarily on the surface because the tardigrade itself is also just a few micron in size. It's within, let's say, a fraction of the volume of the tardigrade.
But how do you know? How do you know that's the fraction or which fraction?
So we have done, let's say, simulations to actually see, let's say, using finite element software to see or to simulate where, let's say, the electric field of the superconducting qubit couples to the tardigrade. And there we actually have, let's say, seen that, let's say, a small volume of the tardigrade close to the surface is, let's say, mostly interacting or strongly interacting with the transform qubit.
So is it possible that so basically the deduction comes from the simulation, right? And then I suppose that you compare the simulations output with the overall, let's say, the frequency that you measure in the frequency measured, and then deduce that it is entangled. Part of it is entangled. So two questions. So basically, that if I have, it has nothing to do with the special, the composition of the tardigrade. It has to do with, let's say, uh, the the low temperature that presumably that all the atoms are in their ground state. So if I replace it with something else, that the inanimate or anything else would get the same result, right? And so that's the first question. And second question, it is it possible to any other, because this is from simulation, right? So not direct, not coming from the direct measurement. Is it possible to I think the simulation, what kind of simulation is it? Is it the density function theory or some other models? And then I guess third question is it possible to produce the same result with, say, a classical system? The yeah. So these are the three questions right now. Thank you.
Yes, yes. Okay, let me try to answer that. Maybe the first question. So in fact, there's let's say for our purposes, or let's say at the end of the day, doing, let's say, the same experiment with let's say another material instead of, let's say, the tardigrade and something else, which has, let's say, the same plausibility as the tardigrade, would actually give, let's say, very similar results.
Let's say there's, let's say, for, let's say, seeing the, let's say, the shift in the frequency of the qubit, which determines the coupling. It doesn't matter if it's alive or dead. Let's say one could even, let's say, think, let's say, having a tardigrade there, a piece of dust would, let's say, give, let's say, a similar shift. What we could do in the experiment is, let's say, only see the, let's say, how the, let's say, tardigrade has acted on the, let's say, qubit itself by looking actually, let's say, at the shift. We were not able to measure the tardigrade on its own. If this is, let's say, sort of a, let's say, entangled state directly, maybe Vlatko, do you want to join in here?
Yes, I think there is one. So I think this question of the crucial question there is, as you said, is whether there is a kind of what we would call a semi-classical model of this. And let me say what this would mean. It would mean that the qubit is treated quantum mechanically in the model, but the tardigrade is kind of modeled as a classical effect on top of this. And of course, we do this all the time in physics. When you look at, for instance, a photon propagating through a material, a dielectric material, you will say that the dielectric is effectively a classical medium that changes the refractive index And basically the phase that the photon picks up as it propagates is somehow explained in this way, in a classical way. So the photon is quantum, the dielectric is treated classically, and that's why we call it semi-classical. You could, of course, have a description like that in this case. You could think of the tardigrade as a kind of classical system, which modifies the energy structure of the qubit. However, one should, and of course, if you want to really argue against this view, if you want to experimentally challenge this, then you must do what we were already saying. You must somehow be able to zoom into the tardigrade and measure directly these tardigrade degrees of freedom that respond to the qubit. You know, this induced dipole that gets induced in the tardigrade because you create it inside the qubit. We haven't done that. So to really close this kind of loophole, if you like, and this semi-classical description, you really must be able to address both of these systems independently, each of them really independently. However, having said this, one has to remember that these semi-classical descriptions are frequently inconsistent from all sorts of other perspectives. For instance, it's very hard to account for certain things like the law of conservation of energy if you have a hybrid system where half of the system is classical and the other system is quantum. Um, and so because of all sorts of other what you would call possibly more circumstantial kind of evidence, it's not a direct evidence like measuring the tardigrade directly, but somehow it's indirect evidence that you can use in order to really say that the only consistent way of treating these things is in fact to really quantumly treat both the tardigrade and the qubit, in which case it's obviously an inescapable conclusion that something inside the tardigrade, and we don't really know how large this is, spatially speaking, how many degrees of freedom are involved, but something inside the tardigrade really is quantum and responds in a quantum mechanical way by getting entangled to the tardigrade. But certainly the evidence as it stands, it's not completely watertight, and these are obviously the things that I think would be the first things we would like to improve and probe better. I think the first question really is, what are the relevant degrees of freedom that respond in this case? How big is this? How macroscopic is this superposition that we are talking about? And while we understand this really well when it comes to the superconducting qubit, we really have, I think, very little understanding what this could be inside the tardigrade. And I think that's kind of the key question as I see it.
Yeah, so and also the number of degree of freedom there is a question, right? The
Yes.
huge number degree of freedom. Yeah, and so it's kind of hard to pinpoint the which is entangled, which is not, or exact, or whether it is.
Yes.
Yeah, and then what about the first question? Is the if you I didn't hear. I think it was mentioned, it was addressed, but I wasn't sure. So the if you replace it with something else, it would uh, it would work as not. I mean, not work as well, but it will have you will have the same response. Would that be yes or no?
Yes, I think you're right. This comes up frequently in many of these discussions when I go to conferences on quantum biology. This question comes up very frequently, and I think it's related to whether this feature would really be somehow important or even intrinsic for the living system. And I think you're right that an inanimate object would frequently respond very similarly. You could think of, like I said, an ordinary dielectric as modifying the refractive index in much the same way, so that the frequency shifts that you observe ultimately are comparable, in fact, to what Reiner observed in the experiment. So certainly you could think of non-living systems producing the same effect. And I think this too is a very interesting question, really. Are there some quantum effects that are really somehow crucial to biological functionality? Do they only occur in some biological scenarios and things like that? It seems to me that this particular experiment, an inanimate object would do the same. And that's a very good point.
Okay. What about the second question? I was just curious what model you're using to simulate. Is it like, say, density function theory model or something else? Or you just, I suppose you're not actually simulating all that number of harmonic oscillators, just
That's right.
maybe some approximation. Okay.
Yes, it's a very simple model, exactly what you said. Because I think I've written a bit more extensively about it. I have a couple of pages, a note that I put on archive a few weeks ago. And I kind of try to offer models with an increasing degree of complexity to take more and more degrees of freedom into account. But as you actually pointed out, it never goes to the degree of detail that you need, that you would use maybe if you were really doing some complicated chemistry simulation. So certainly density functional theory and things like that don't really need to be used here. It would be an overkill given that experimentally really what you are ultimately measuring are the frequency shifts, and you can really account for them in a much simpler way, namely through coupled harmonic oscillators, and then of course you can add more harmonic oscillators to represent other degrees of freedom. But I think nothing really more than that is required in this case. Very similar to a typical, probably thinking of a dielectric, and dielectric affecting the refractive index is maybe the right model, and the complexity to think in this case.
Okay, so then how many, what is the number of harmonic oscillators, a couple of harmonic oscillators do you use in the simulation?
That's completely open, actually, because again, given that you have to account only for the frequency shift, even a simple model with two coupled harmonic oscillators will do the job. One representing the superconducting qubit, and the other one representing collective, I would say, degrees of freedom of the tardigrade. I mean, even incidentally, for the qubit, you don't really need a harmonic. First of all, it's an unharmonic oscillator, and you are using the lowest two energy levels. So it's really just a two-level system coupled to a harmonic oscillator. So much like what one would call a kind of James Cummings model. And this is simply because we have this big uncertainty about the kind of the exact the exact properties of the molecules that would be responsible for this in the tardigrade. If you did know these molecules more, you could take into account all the vibrational degrees of freedom, not just electronic and so on. And then you'd be talking about a much more complex system. So really, the number of harmonic oscillators would depend on how many of these modes are engaged in your response. And at the moment, we really have no way of knowing this.
Yeah, no, the qubit, I'm not talking about the qubit. Yeah, as you said, the qubit can be uh, simulated very simply. There's like the basically uh, two-level system. And
That's it.
I was just worrying about, yeah, I was just worrying about the particles in the tardigrade. And I'm just thinking that whether if you, even with the harmonic oscillators simulation, approximation, does the increment of the degree of freedom, how does that affect the results of your simulation? When you yeah, increase the degree of freedom.
I don't think you could see that. Maybe I can check with Rainer. Just maybe he can tell us in a bit more detail. But I think this is very hard, given that what you're ultimately measuring, at least in this first experiment, are these shifts that you get from the presence of the tardigrade. This second experiment, of course, I think builds on already this hybrid system. So I don't think it's relevant at all when the second qubit gets engaged. I mean, Rainer, would you say that it's difficult to say more from the experiment, right?
Yes. Well, okay. Maybe it's sort of one could see a hint. I mean, of course, you have, let's say, too many degrees of freedom, the whole system becomes the environment. So meaning actually that the coherence time of the system will decrease because it's coupled to effectively the environment. So I would say actually we have, let's say, a finite amount of degrees of the freedom inside the system. And with this, actually, this we have seen that the, let's say, T1 time of the system did not actually change, of the coupled system did not change significantly. That would be, let's say, another way to, let's say, look at, let's say, the amount of, let's say, degrees of the freedom we actually have within inside the coupled of the system.
Yeah, so the degree of you're referring to the degree of freedom in the tardigrade, right? Or the modeling for modeling the tardigrade, right?
Right. So let's say for, let's say, modeling the tardigrade and seeing actually the shift in the, or let's say for modeling the shift in the frequency which we actually would expect, what one can just use, let's say, normal electromagnetic software, finite element methods to actually look at the, let's say, use a medium with a certain probability, and then actually look at, let's say, the frequency shift one would actually observe in the experiments. So you could actually model to a certain extent the, let's say, response of a direct medium directly using normal software you usually use for modeling electromagnetics.
Okay.
Yeah, does anyone else maybe want to raise a question? Just checking. Please flash your mic when you have a question, please.
Hello. Yeah, this is I think very interesting because quantum and biology together. You know, there's I think the frontier of science, right? So my question is the uh, w- um, on the, I mean, probably follow up from Hansen. I mean, I was actually listening and not completely understanding, but I just wanted to check that. So at this point, the I mean the experiments and so there are two qubits, and there's a frozen animal, tardigrade, in between, and you were able to see changes to the original entangled states. And is that correct? I mean, I haven't read the paper yet.
Yes, that's describes it, the experiment exactly. So we had let's say this experiment has had these two stages. First, we had, let's say, the qubit, put the tardigrade on in the TAN state, which, as was also pointed out by Hansen, let's say could be also, well, was a ametabolic, it was an ametabolic state, so very much close to a non-living state. This was coupled to the qubit, where you see, let's say, response from the qubit on this. And then let's say this coupled state, or this dress state, one could say, was covered again with a second qubit to actually make, let's say, base state of this coupled qubit tardigrade system with a second transform qubit.
So I guess two questions. I mean, one of them is after you raise the temperature and the tardigrade just recovered and nothing changed, right? So it's very well alive. And the other is the evidence. I mean, at this point, there's completely no doubts that is the. I mean, yeah. I guess Hansen asked if you substitute that tardigrade with some other objects in similar geometrical aspects. Have you tried that already or not?
Well, so to the first part of your question, right after we have, let's say, brought back the tardigrade to the normal ambient conditions, so room temperature and normal pressure, the tardigrade changed from the TAN state to the, let's say, normal living state and could actually see it was living again. Having said this, maybe I should also emphasize that it was, let's say, very crucial the way how we actually brought back the tardigrade from, let's say, its cryptobiosis state to the ambient conditions again. So we have to actually increase the temperatures or the temperature very slowly, and also what was seemed to be also very important is to actually do the same with the pressure. So actually increase the pressure extremely slowly. It doesn't matter so much that, let's say, in the range, if you're, let's say, 10 to minus 8 or so millibar, it doesn't matter if you increase from 10 to minus 8 to 10 to minus 4 fast. But let's say in the range of about 10 to minus 3 to normal room temperature, this has to be increased slowly. Also for the temperatures itself, at, let's say, yeah, probably something, let's say, if you're at, let's say, millikelvin and you go to, let's say, minus, or let's say to maybe something like 200 Kelvin, the gradient doesn't really matter. It actually matters more when actually, yeah, in the range when water starts to melt again. Okay, that's the answer to the first question. The second question was again using, let's say, a living system and, let's say, a dead system. If we would see the same response on the qubit, which we would actually see if they have the same, let's say, polarizability in this case.
I see. So there's actually, I noticed I followed a earlier experiments by Kohler and etc. Et al. On the macroscopic, they call it, entanglement with, I think, oscillating drums. And I mean, in macroscopic scale, and they cited Simon Dorn's criterion for to say that it's entangled. Is that relevant here, or do you consider that's interesting work relevant to your future research.
So let's say the work is of course extremely interesting and relevant. It is, let's say, what we see to a certain extent is it's not really a Schrödinger cat experiment where you have, let's say, a very macroscopic system entangled. Like let's say this, for instance, mechanical oscillators. It's actually the, let's say, molecules inside the tardigrades itself. Or let's say or let's say a collection of molecules inside the tardigrade. Right.
Yeah, so maybe, you know, further with a very clever and careful experiment, you can somehow go into the macroscopic object here and see some type of time and spatial, you know, how to say, scale. I mean, kind of a phenomena, and pointing to that's actually a microscopic entanglement. I don't know. I mean, I'm just, you know, bringing ideas. Yeah.
Yes, yes, I agree. I mean, so again, so this are, let's say, maybe what we have shown is just the starting step for actually the more exciting experiments, where we, let's say, can go to, let's say, bigger systems, and let's also look at macroscopic entanglement of, let's say, some maybe macroscopic degrees of freedom. So, for instance, motion would be something to look at as well. But this you can't do with, let's say, swinging in qubits. For this, you may need some micro oscillators. But yeah, this is could be done, let's say, in future experiments or future work. Rajko, do you want to add there a few words? I think maybe there was also this experience with the bacteria.
Yes, yes, I think my friend Dave Coles, actually, who is in Sheffield now, but he was at Oxford with me at the time. He did an experiment with a living bacterium inside an optical cavity and coupled, also observing frequency shifts that you would normally see in a certain place when the bacterium was in a different environment. Now these could be shifted because of the strong coupling of the bacterium to the light inside the cavity. They are certainly interesting. I mean, they are kind of along similar lines to what we've been discussing. Of course, the bacterium is far simpler than a tardigrade. The only the difference here, of course, is that it was done at room temperature, and you could really see that the bacterium was alive during this experiment. However, even for an experiment of this kind, I think it's still correct that you could mimic this with an inanimate object. I don't think it was crucial, really, that the experiment was done with a living system. It just was impressive to us physicists, certainly, that a biological system can actually be manipulated to be in this kind of quantum mechanical state. The only thing I wanted to add to this various macros, by the way, our field is full of people making all sorts of macroscopic entangled states. One just has to be a little bit careful when you use the word macroscopic, because even though sometimes people may use systems that look big, when you really look into it, what happens is that the degrees of freedom that do contribute to this entanglement end up being very small. Effectively, you are creating 2 qubits worth of entanglement even though your systems might be far bigger, physically speaking, their response comes from a far simpler subsystem, if you like. So, people claim, for instance, to entangle two diamonds. And of course, they didn't entangle the whole diamond. It's not that the diamond is in a superposition of being in two different locations at the same time. What happens instead is that one atom inside this, in fact, one spin, if you like, inside this diamond couples to another spin, another qubit inside another diamond. So I think one has to be very careful um, calling some of these things macroscopic. I think we're still exploring what that means, and we are kind of making steps in the macroscopic direction. But it's possibly fair to say that no one has really made a genuine macroscopic. I mean, of course, there is no clear cutoff. How many atoms should contribute to a superposition so that we call this macroscopic? There is no answer to this question. But I think genuinely no one has really created a macroscopic. So, for instance, a bacterium being superposed across two physical locations at the same time. This would be impressive. It's even questionable whether this is possible, by the way. Maybe this is too complex to ever be able to execute physically. But I think these are very interesting questions. So the experiments we are all discussing and the ones you mentioned are all to do with the subsets of these systems that actually do contribute to the quantum superposition, but it's not clear that any of them should really be called, genuinely macroscopic.
I suppose that
Thank you for. Yeah, sir. Quickly. Thank you for both great answers. So, when you mentioned the indeterministic state, I mean, currently still, I mean, on the question of macroscopic entanglement,
Yes.
I'm just curious. Because I'm definitely a layperson and. Because there is, I mean, for example, in Kotler's paper referring to the Simon Duan criterion, if I understand, I mean, just very brief reading has to do with the matrices, the matrix of the system. Whether it shows us it's entangled or not. Is this still that criterion itself is still in a research stage, or it's
Oh,
well accepted?
good question. Good question. The criterion is mathematically perfectly sound. It's a good criterion. Of course, people are investigating even theoretically different criteria. We have all sorts of criteria that would work in the macroscopic limit. You can measure things like heat capacity, magnetic response, magnetic susceptibility. All of these are nice entanglement witnesses. And the one you mentioned by Duani, in fact, applies to systems that are as simple as two harmonic oscillators. And in fact, that's perfectly suitable even to the experiment that Rainer did. So it's nice that you mentioned that criterion. It is possible to use it in our context as well, but one just has to still bear in mind that whether this is macroscopic, or it can certainly be applied to a macroscopic system, but it can also be applied to a very simple system that only has two harmonic oscillators. The thing that's crucial really is somehow to be able to measure corresponding properties of each of these harmonic oscillator. So you would have to measure something like the position and the momentum of each of them, and then look at the correlations. And the strength of these correlations tells you really whether the state is genuinely quantum entangled or not. So I think that criterion is great. It's certainly applicable to our situation. But it doesn't necessarily tell you whether something is macroscopic or not. It just tells you if it's entangled or not.
Vlatko and Rainer, I have a question for you regarding your beautiful paper, which I very much enjoyed reading, and potentially someone else has already asked this. but I just joined. Unfortunately, I just saw that you were having this event now. So I wanted to ask in what sense the tardigrade is entangled with the qubit. So it looks like it could also just be a dielectric, which is basically shifting the resonance of your high Q resonator. So I come from optics. I have a completely different background. So my question might be completely off here, but maybe you could explain to someone with like my background how yeah, this is entangled rather than basically the tardigrade acting as a dielectric shifting the resonance.
Rainer, do you want to have a go? I think it's something we touched on actually kind of 5 or 10 minutes ago. And we had a
Oh, I'm so
discussion
sorry
about it.
about that. I'll listen to the replay in that case.
I think the brief answer is that certainly there is a range of models that we can use, and I think I talked a little bit about what you would call a semi-classical model, where you treat the qubit quantumly and you treat the tardigrade as a dielectric. And it's certainly possible to do that. I think that there is no question about it. And given that if you can experimentally really only extract a very small number of parameters, then definitely your model doesn't need to be much more complicated to account for that. What's problematic with semi-classical models is that they're inconsistent frequently for all sorts of other reasons. And I don't want to go too much into that. Some of these failures are kind of known, and however, to really conclude, if you want to experimentally conclusively show that there is entanglement between the two, it would require something that we don't have in Reiner's experiment, which is a kind of a degree of control to be able to measure the tardigrade directly rather than just by a coupling to the qubit. So some kind of probe that would go into the electronic degrees of freedom of the tardigrade and measure some kind of complementary observables independently to the measurements of the qubit would certainly be necessary in order to close this and to kind of clinch the argument that this is entangled. So we have what one would call kind of more circumstantial evidence that this is the case, but not a more direct experimental evidence. I don't
Oh,
know if
thank you.
Rainer wants
Yeah,
to.
no, that actually makes a lot of sense because just from a simple argumentation, I could also imagine that this kind of resonance shift would just be a result of, yeah, just there being a clump of dielectric essentially. But yeah, adding like a more direct probe in the tardigrade would actually be great. Yeah.
Yes, I think so. I think it's necessary, and that seems like a kind of one of the following kind of logical steps to undertake in the future.
Okay, thanks a lot for your answer. Thank you.
Thanks a lot.
So with regard to the diamond example you mentioned, I suppose that the two atoms in in entanglement are pre-located. Is that right? And then that's also related to the previous question I asked, which is repeated by Xinghui. The if the if we know that there's only very small part of the tardigrade is in supposedly suspected to be in entanglement, and then you don't know which one it is, even if you have a probe, how are you going to probe the tardigrade? Presumably, you have to either establish a lot of probes or have to kind of move the probe around. Yeah. Okay. So this is my question so far.
Yes, very good point. I think in order to be able to probe the tardigrade more directly, you would certainly need to understand which degrees of freedom are relevant. And then on top of it, you would have to understand how to interact with them, how to probe them. So this is crucial. it would, on the positive side, once you know that, it doesn't necessarily imply that you really have to measure each of these constituents at the microscopic level. As we heard, there are criteria that actually could be macroscopic. They come from kind of thermodynamics as a collective response, and maybe all it would take is to look at the collective electric response of whatever the molecules are that are engaged in this kind of state, entangled state with the qubit. But I think it's certainly important first to identify that before we can then see how to couple to it independently and how to probe it. The difficulty, as I see it, other than identifying this, is actually to find these degrees of freedom that we can measure. So strictly speaking, you need kind of two complementary degrees of freedom of the qubit, which is easy, of course, because the qubit can be manipulated in all possible ways, quantum ways. But then you equally need two kind of complementary quantum degrees of freedom inside that tardigrade, and you need to be able to measure them. And the question is, can we identify that? Why is this a question? Because if this system is very complicated, and I think Rainer touched on that as well, the question is whether it's too noisy for certain measurements to be really performed accurately or quickly enough. It's always to do with the relative times, of course, how quick your measurement is versus how quick the noise is. And so this has to be, of course, first understood and studied carefully before we really choose the right kind of measurement to make. And so this is a very important point that you are raising.
Okay, thank you. So, coming back to the previous example that you mentioned, which has nothing to do with your experiment, you said there were bacterias also entangled
Yes.
with. Yeah, but then you also mentioned that it is done in room temperature. So how now in your experiment, it is plausible because uh, the temperature is close to absolute zero, and then 0 Kelvin, and then so presumably there uh, almost all of the atomic states are in the ground state. But then how do you explain the bacteria that claim that bacteria is in the entanglement because it's in the room temperature, and then you can have lots of state and so many degrees freedom, and then the energy that distribution, all that. How do they? I mean, not you. How do they can claim it's in entanglement?
Yes, I think the claim there is because you are studying the electronic transitions inside the bacterium, whose energies are well above uh, the, the room temperature energy. You know, the KT, the Boltzmann constant times the temperature. And actually, this is true for ordinary atoms as well. All of the atoms at room temperature are effectively in the ground state. The reason being that the thermal energy at room temperature is about 40 times, 40 times smaller than the energy that is needed to excite the atom from the ground state to the first electronic excited state. So even room temperature, effectively, if you're looking at the right transitions, is in fact not sufficient to get things away from the ground state. In Reiner's experiment, and I think Reiner can maybe say a bit more about it, we are talking about superconducting qubits, and the energy, the relevant energies that we are discussing that are very different. They're very smaller, and you really need to operate at a very low temperature to make sure that you are really in the ground state and you're not exciting the system. But that's because the energy gap is much smaller than the energy gap that was relevant inside the bacterium. I don't know if Rainer, you want to say a bit more about this.
Yeah, absolutely. You're absolutely right. So let's say, yeah. So if you want to, let's say, mentioned before, bring, let's say, living or some system and tangle this with a quantum mechanical system. Then let's say if you're going to, let's say if you're going for superconducting qubits, for example, they're working at, let's say, typical frequencies or energies of gigahertz. So meaning actually the environment here where the system is operating has to be, or yeah, the energies of the environment has to be lower than h bar omega with a frequency omega of gigahertz. So meaning this will be in the range of millikelvin, where, let's say, the peak in the black body radiation will be below gigahertz. The let's say experiments with the bacteria is actually went, as Vlatko was saying, to, let's say, higher energies. So they are actually the energies which are relevant are optical frequencies, which can actually being used inside, let's say, ambient environment at, let's say, 300 Kelvin room temperature. And also the time scales for this are much faster, let's say, at room temperatures. Whereas for the superconducting part, the time scales are in terms of yeah, microseconds to, let's say, roughly 10 microseconds. So it's, let's say, depending on which energy range you are, you have to change, let's say, or you have to change the timescales of your quantum system, and also the, let's say, energy spacing of the quantum system.
I see. So basically, the room temperature bacteria, they using say laser that the energy excitation energy difference is far larger. The the different energy level is far larger than the room temperature energy can provide, right?
Exactly.
Okay. But I guess that you have to put it like the amount of energy in. Okay, so it's a case of probability, or yeah, I have to look at the numbers, right? If the it's a it's a question of probability whether the bacteria, all the molecules are in the low small probability case that all the energy, the thermal energy that I call concentrate at one moment into the excitation of one atom or something. That's possible, but low probability, I guess.
Yeah,
If your energy
well,
excitation energy is high enough. Okay, go ahead. Sorry.
yeah, yes, exactly. So to be fair, it's still, let's say, a thermal state. However, the probability of the excitation to an excited state is extremely low. That's how I would describe this. But that's coming, let's say, a bit from the background of atomic physics and also, let's say, laser cooling, how to cool actually atoms down. We can actually cool, let's say, atoms to the, let's say, to ground states of the traps. If you're using normal laser cooling, we get still a thermal state with, let's say, a very low probability, let's say, of exciting higher vibrational states. So again, it's a thermal state, but the probability is very low that that the system is excited in a higher state. That's exactly what you have mentioned.
Yeah, yeah. Thank you very much.
Okay.
I had a quick question. This is Daniels. What happens when they come to temperature too rapidly in terms of when you're rewarming them?
So when we, let's say, increase the temperature and the pressure too rapidly, what we actually have seen is that, the tardigrade, that's just by evidence. What we have seen is that the tardigrade will not, leave its stun state. In fact, actually, what we have seen is that the tardigrade was somehow disintegrating. Why is this exactly happening when you're doing it too rapidly? I'm not, let's say, 100% sure. I actually don't know because I'm not a biologist. Yes, but let's say this is from the evidence that if this is done, let's say, too quickly, heating up and increasing the pressure, they won't survive.
That was certainly what I expected to hear. I was thinking more of like a situation where it was not under pressure, but just temperature differentials. And what I expected to hear was that if it was in a crystalline form, I know that some tardigrades, I think the ones that have been found off-world in outer space, they assume a crystalline form at some point. And obviously, if you expand that form too rapidly, it's going to cause chaotic failure in the reconstitution of their life.
Yes, yes. So let's say we have used let's say a tardigrade species of tardigrade found in Denmark, which has or was shown to be, let's say, fairly robust in, let's say, if we expose it to harsh environments, and let's say also bring it back to life. Yes, and the, let's say, as you have said, that if you, let's say, change the environmental condition to, let's say, be more adverse, the tardigrade goes into this TAN state, which is a state where, let's say, the tardigrade expels all its, let's say, liquid or water content. And that occurs, or that decreases the volume. And apparently in this state, it's let's say it's a very robust state, which is able to sustain in this, let's say, harsh environment. So inside the tardigrade, most likely there's let's say there's no liquids anymore, which can actually freeze. Because if it's starting to freeze and crystals are forming within the tardigrade, this can actually also destroy the organism.
A delicate procedure, to be sure.
Yes, I have a question. I was wondering, is there the implication maybe? So my understanding, I came late to the room, but it seems from what Vlatko said is that we have a large quantum effect on a very small part of a larger object, which is the tardigrade. So you would assume, therefore, that it really didn't have any effect on keeping it in cryptobiosis. But then there was the statement in the thing I read here at the link is that it actually broke a record. So is the possible implication is that even though there should be just a small effect on, or a big quantum effect on a very small part of the larger object, the tardigrade, is the implication that maybe something else happened, which is why the tardigrade was able to remain this way in breaking records, or is that the possible implication?
Okay, maybe I jump in, maybe but can add to this later. So the measurements which we have done with the tardigrade and the sogenannten qubit have no implication on the state of the tardigrade after the measurements. So meaning, let's say this record which we have done, let's say having the tardigrade inside the dilution refrigerator for 420 hours at this very, very low pressures, the tardigrade would be capable of doing this also without doing these experiments which we have done.
So it just happened to be a record, probably
Yes,
created by the
exactly.
conditions, not that some implication of something else happening. Okay, thank you.
Exactly. Yes.
It was
However, it would be very interesting, maybe in some future experiments, to see if there's some manipulation, let's say within microwave frequencies on the tardigrade, that this maybe has some effects later on the tardigrade when warming up. But we are, let's say, far away from this situation right now.
Yeah, if anyone else has a question, please go ahead. But then I wanted in the meantime ask if you are still fine answering questions or if you have to go, because you've been talking for like over 90 minutes. So
Katerina, I do have a not question, but just a statement, because I recognize the name of someone on the stage, and so, and we're actually, is it all right if I mention a club of ours later on today? Yes, so hi, Vlatko. I'm a friend here with Keith and also a guy named Andrei Vajnov on the Clubhouse, and we're actually we're doing we have a club called Gödel's Parallax, and I think a lot of people are interested. We're actually doing the book, The Science. We have a kind of a book club, and we're actually discussing the science of can and can't tonight. And I saw your name had been mentioned in there as an acknowledgement, so take a moment here to promote that. And if you were interested in joining us, that would be great, as well as everyone else on the stage who probably has interest in these kinds of topics. So we are doing that book tonight.
All I can add is a great book. I don't know what the only contribution to that book from my side of things is my drawings. Actually, they're not that good, but I think Chiara was very nice to actually put them in the book, and that's it. But it's a great book.
Yes, I've been enjoying it, and we're gonna be talking about it tonight.
Great.
I wanted to add, Eric and I, we have been talking to, we will have a lot of guest invited speakers, scientists, and we have been having them. We thought maybe to do kind of a day on Clubhouse because, most people that we invite, they just join. And we thought maybe they could have a day on Clubhouse and go maybe not to just one club, but then to different ones. Maybe Ethan, we can talk about that idea. So when the speakers come to Clubhouse, that they will have like reached the biggest audience possible by going around in clubs that are interesting for them.
Sure, I'd be happy to discuss it. Or Andre, Keith, also. I also should mention it's it's an informal book club with the odd rule that you don't actually have to have read the book to participate. Always, because we know a lot of book clubs fail because not everyone is up to date exactly on the reading. So we've made sort of we started off with just the three of us, but we've enjoyed having other people join on as well. And we did that just because the pragmatic reality is that hardly everyone in book clubs that I've experienced or talked with people ever have enough people who fully read the book, or they've read half of it, or some people have read it 6 months earlier. So we figured we'd make a club and give an experiment to a book club that didn't quite read the book, where some people have and some people haven't. But it's actually turned out to work out quite well.
Yeah, I should come. Thank you for mentioning it. We appreciate it. Yeah, if anyone else has questions, I know that there are a few on the stage that didn't have a chance yet to speak. So, yeah, please flash your mic, go ahead, and yeah, ask your question.
Just quickly, which is it again? Sorry.
I'm sorry, were you asking me a question, Hanson?
Yeah, just quickly, what's the title of the book again?
The book is called The Science of Can and Can't by Chiara, I don't know how to pronounce her name exactly, Chiara Marletto. And that is the book we're doing tonight in Gödel's Paradox, the club Gödel's Paradox.
Okay.
Parallax, sorry, Gödel's Parallax. And
I have a very basic question regarding quantum biology. Maybe somebody already asked this because I arrived a little bit late to the presentation today. But so I recently came across an article that claimed that like some like processes of the cell membrane were better described by quantum information. I think is by Michael Levine or Levine. And I just wanted to know if maybe there's been recently any discovery. I'm not really an expert on this topic, of any like natural process within biology at let's say uh, just as you guys mentioned, it's difficult to talk about macro scale, but perhaps a biological process that um, requires like let's say a lot of machinery behind it that uses any quantum effect, but let's say that has been operating within the natural realm, not engineered by humans.
my understanding is, no, but it's always the sort of holy grail that I think people want to hand wave and make everything quantum consciousness, So, I mean, the non-trivial quantum stuff, I think my understanding has been that the most likely one that people thought would be in odor detection and taste receptors, but that keeps not really popping up. And of course, like Penrose and people have kind of conjectured these phenomena, the non-trivial quantum phenomena, and somehow linking them to some sort of important part of either living systems or intelligence or cognition. But I don't think, it's ever actually been shown anywhere. But I think that's, it's the air of mystery that I think Andre talked about. He called the preservation of mystery. So I don't think there is one. Or the microtubules. I still don't think they've really showed anything there. Prior to that, Penrose in his first book thought it might be going on somewhere else, but it was just a placeholder. But this, Keith and I have talked about this a lot, but this has been something that's been done since the early days. The first being one of Heinz. I think it was during the modern physicists. He went on to become a biologist, but I can't remember his name, but he was the first to kind of posit maybe some kind of thing like this was happening in living systems, but he quickly abandoned it, and then continued to do other great things in biology. But people have been circling that ever since. I think there's value to it, but after we've understand the non-quantum, I think there'll be many non-quantum explanations for knowledge and intelligence and living or intelligent systems. And then they'll be supercharged by understanding how to manipulate quantum information, so that it would be like the flight is happening naturally in birds and there are physical principles, but the jet engine is what got created for super fast flight, but they're not really connected directly. So I think we'll have AI and maybe quantum systems that can do things faster and better, but it won't be per se needed to make that happen, which is, I think, the link to trying to find some non-trivial biological system that exhibits like superposition or entanglement.
I would say
Uh,
I would say that the Penrose model has that rotating molecule, and that kind of resembles like a hashing algorithm, in my opinion. So there could be some sort of hashed entanglement. So it's not necessarily that all the molecules and particles are exhibiting this kind of behavior, but you could distribute it out in a network, and therefore, when you have like a protein and it bends a little bit from one state to another, that might represent, for example, one state of entanglement versus another. So, but I'll send you that paper, Ethan. It would be awesome to talk about.
Yeah, I also want to tell Keith that since he was talking about the work of Michael Levine, he will be here. He will be a speaker on February 24. Is that February 24, Katerina?
Uh,
He will be,
yeah.
yeah? Yeah, he will be speaking about his work. I think it also involves consciousness because in our other club, he has been a guest together with Josh Bongard, who is doing evolutionary computing because of the talks on the xenobots. But he has other researches on electrophysiology. And actually, I think there was another paper which was released, and he was working with the physicists to tie up his research with quantum mechanics. So yeah, that's another paper that he will talk about maybe in the future, but he's coming here very soon. And yeah, I hope you can attend that session so that you have a chance to talk to him. Because we might be talking about electrophysiology and also consciousness. So yeah, please attend that session.
Yeah.
And yeah, I just remembered the name. It's Max Delbruck, who was he was a modern physicist. I think he was Heisenberg's or one of those guys, graduate students, but he went on to be a biologist. But he was the first to sort of think about trying to find some sort of modern physics as being an explanation of living things or consciousness. So he had a book called Mind for Matter, and he went on to win a Nobel Prize in physiology. But he was kind of the first to bring up all the quantum consciousness kind of possibilities, but he also kind of put them to bed as well.
Yeah, just to say, it's February 23rd at 4 p.m., just like today. Michael Levine will be here to share his one of his newest. They are still in, the paper is still in bioarchive. It's about consciousness and the mind, research and theories. It's I think it will be really interesting. If they were here before to talk about the xenobots that can self-replicate. So yeah, if you have any questions about this work, come back. Yeah, Vlatko and Rainer, you have been here now almost 2 hours. So I wanted to check in with you. How much time you have, or if we should close the room. And please let me know.
Yeah, I think I have to probably decouple in about 5-10 minutes. But I mean, please do continue. Don't let that disrupt the flow if you have it. I certainly enjoyed the discussion. I'm happy to answer questions by email or any other means actually about this topic. And hopefully next time we'll be able to tell you more about the future experiments as well.
Oh yeah, that would be amazing. Please come back and keep us updated. It's we are all
Thanks
very,
a lot.
very interested to hear. And always come back to Clubhouse, to our club, and join the discussions. We will have upcoming rooms. The next room is tonight at 10 p.m. EST, and Dr. Field will talk about his new invention, that he did with his company. They developed a wearable device that does non-invasive, very precise optical brain imaging, which is very excited because usually to do this precise brain imaging, people have to lay down very still, lay in this tube, and he developed like a device that you can wear on your head and do optical brain imaging. So that's really very exciting. So yeah, come back later and follow the club. We have more rooms coming up with guest speakers. As Cecile already mentioned, on February 22nd, next Tuesday, we'll have Dr. Jones. He will talk about stochastic microbiome assembly, and that it depends on context. So I heard a lot that people are interested in the microbiome. So there will have a chance to talk with a researcher that works on the microbiome. Yeah, and then Dr. Levine will come back, and Dr. Kaufer, she works on anxiety and PTSD research, and she saw differences in myelin in people that have anxiety and PTSD compared to a healthy individual. So, yeah, please come back, but go ahead and continue with the discussion. It's very nice.
Yeah, I
Can
just
I have
wanted
a
to
question?
mention the paper I mentioned from Michael Levin is called, the name is Metabolic Limits on Classical Information Processing by Biological Cells. So he uses actually some quantum information to analyze the decoherence in the surroundings of the cell membrane. And yeah, I think it's pretty interesting. Yeah.
I just want to mention also that his work on consciousness, actually, he wanted to bring in here Daniel Dennett because he is a collaborator in his work on the point of philosophy. But Daniel Dennett is not available. So, yeah, he's just coming in. So, I think that this coming talk would be very interesting.
Can I ask a question?
Yes, please go ahead.
I see. Oh, okay. Rainer's still here. Yeah. So, Professor Weller mentioned the future experiments. Yeah, I was wondering, what the could you share with us? I mean, some preview. You know what? So, is it very difficult if you have the tardigrade to wake up slowly, properly, while still maintaining the entanglements? And because they can do that with bacteria, probably it's doable. But probably, would you share? You know, what would be the challenges if that, you know, is relevant, or I mean, what type of direction that you're thinking of at this point?
Okay, so let's say waking up the tardigrade while still maintaining the entanglement is something we cannot do, because again, this coupling to the quantum mechanics systems only happened at this, let's say, very low energy environment. So in this midi-Kelvin stage. So if we leave this energies, then let's say this quantum system will be masked by ambient noise. So it's not possible to see it. For the bacteria, the difference was again that the energies are much higher. So we had there this light energies, given by light, the frequencies, which are, let's say, large compared to, let's say, the black body radiation at 300 Kelvin, for example. Future experiments which we are, let's say, envision and thinking of, we still have to see if we can actually conduct them, is actually to find, it's inside the tardigrade, the that's a part of the tardigrade which interacted with the qubit and able to actually, or try to see if you're able to actually to probe this part, let's say for example in frequency space. That's actually what we envision to do actually in the future, to really then do measurements independently on the tardigrade and let's say the qubit system itself to um, then let's say be able to also give a bound on the, let's say, one could say amount of entanglement of the tardigrade with a qubit. I hope this answered the question.
Yeah, thank you.
So I had a question more on the biology. I really appreciate being here and hearing about some of your future work to really pin down the quantum mechanics side of what is going on. But I'm particularly fascinated because you were able to put them into a really extreme state of tun for much longer than anyone has tried. I was wondering if you had, I know it's outside really your bailiwick, but whether you had any biologists come and want to follow up on this because it is a very interesting field as far as cryobiology, trying to figure out everything from freezing human ova, which is could be nice, to uh, making some nice popsicles for space travel. So
Yes. So let's say the biologist on our team was Nadja Mudberg. She also had helped us with, let's say, or taught us how to handle the tardigrade. And let's say when discussing with her and so on, there was still, it's, if I understand correctly, I'm a physicist, I'm not a biologist, so if I understand correctly, there was still sort of some of the assumption that also in the cryptobiosis state, there was, let's say, no measurable metabolism going on, but maybe there's still some small metabolism going on within the, let's say,
yeah,
within the
yeah.
tun
The
state.
the, the, the
However,
argument seems to be that maybe there's it's slowed down basically about 100 to 1,000% versus entirely frozen.
right,
Uh,
exactly. So, but
yeah,
I
exactly.
think what we could actually show with this experiment, let's say this maybe on the side track, is actually that it is totally came to a standstill. I mean, at
Yeah.
this again at this millikelvin environment, the let's say everything is in their ground state. So one could say, yeah, there's no movement, there's no chemical reaction,
Yep.
there's nothing
No.
going on.
No.
It's
No.
zero.
No. Yeah. Yeah.
So it's
Yeah.
yeah, there's no metabolism at all. That's maybe let's say on the biological side what we could actually show there as well.
Yeah, because it's been tough naturally to find tardigrades that are naturally in tune for a long time. It was previously 2,000 years. I think they found some considerably older ones now. But yeah, that was another. You know, from the biology side, that was something that came out of it that I found really interesting and important. Anyway, yeah, thank you.
Yeah, thanks for the question.
Yeah, if there are no more questions, we don't want to take too much time from Rainer. Maybe he'll just come back. We can arrange for him to come back sometime, and we'll discuss some more, maybe with updates. So yeah, thank you so much for being here and for making this happen with us. We really appreciate you coming and answering all these questions. This was really amazing, and yeah, we want to thank you all for being here.
Absolutely. Thanks actually for inviting me here. I mean, it was a very interesting experience. It was very nice. I enjoyed the discussion very much. The questions were very, really on topic and very, very precise. So I also liked the discussion actually more than, let's say, the earlier 20 minutes presentation. So yeah, I think this format is maybe the future of science. Who knows? Okay, thank you.
Yeah, so I really enjoy it too because you get to read so many different people, and sometimes really interesting ideas and conversations come up that we usually in conferences we don't have the opportunity to. So I agree with you, and yeah, I enjoy it also very much. So yeah, maybe you become also a Clubhouse addict like we are.
Yeah, possibly, exactly. Okay.
It's a natural transition from podcasts, right? So audio signals, as I learned on Clubhouse, actually faster than visual, and allow us to do multitasking. So it is the future. I'm Ragnar. I am with you then on that.
Yeah, I think you're right, Frank. Let's see.
Okay, with that, thank you all very much. See you back in 4 hours, where we have the amazing inventor guest speaker that developed this really groundbreaking brain imaging for humans device. Because if we can move around and do precise brain imaging, that will totally be a game changer. Um, yeah, so please come back, and thanks everyone for attending and asking all these great questions. This was great. So bye, guys. I'll close the room now.
Thanks, Katerina.
Bye.
Bye. Thank you.
Thank you,
Thanks.
Katarina. This has been fantastic. So interesting.
Yeah, glad you. Thank you. Bye.